everyone. Good afternoon. Thanks for coming. Can everyone hear me okay and see my screen?
Yes.
Great. Thank you. All right. Well, I'm really excited to present today, although it looks like I did not update my date on this PowerPoint. I apologize. My name's Allyson Alexander. I'm one of the neurosurgeons here, and Dr. O'Neill and I, as well as Dr. Handler, do the epilepsy surgery here. You've heard a lot about the clinical things that are going on at Children's Colorado over the past couple hours. So I'd like to take a little bit of a different tack now and talk about the some of the research that I do in my lab and why I think that it's important and exciting that we do this. So on this first slide here, what you see on the background is something called a Golgi stain. This Golgi stain was actually invented in the late 1880s by a neuroscientist by the name of Camillo Golgi. It was discovered, as are many scientific things, sort of on accident when he put some silver with some brain tissue and was able to see these individual neurons. So this here is actually neurons staining from some human tissue that we removed during an epilepsy surgery, and you can see the individual brain cells. The cool thing about this technique is that it doesn't stain all of the neurons. The brain is obviously very densely packed with neurons or brain cells, but with this technique, you just get a few of the brain cells stained so that you can see the processes of the cells. So that's just my background slide there. So let me keep going. So the things that I'm going to talk about this afternoon are, first of all, why should we do research on human brain tissue in epilepsy? What are the opportunities that we as scientists can find in working with human tissue? What are the challenges that we can find working with human tissue? And then I'd like to tell you a little bit about the project that my lab has been working on over the last couple of years, using both mouse and human brain. And we study a type of epilepsy called focal cortical dysplasia, which I'll tell you a little bit about. So why should we use human brain tissue to study epilepsy when there are a lot of people out there that work on mice? Well, as you can well imagine, a mouse is not the same as a person. And that may seem trivial and obvious, but it's actually, I don't mean it in a trivial or obvious way. So let's dive a little bit deeper into that. So this is a schematic of a mouse brain. And this blue line down here is 1 centimeter. So mouse brains are pretty small. It contains approximately 70 million neurons, which seems like a lot. And on the right there is just a picture of a mouse brain with some of the neurons stained. So already pretty complicated. However, if we shrink this down to size and then compare it to the size of a human brain, there are actually 86 billion neurons in the human brain compared to the 70 million neurons in the mouse brain. These two pictures of the brain are now to scale. And so you can see just sort of, you can just imagine the complexity and size differences of the human brain just sort of right off the bat. That's obviously not the only difference between the human brain and the mouse brain. One of the other things that you can see, I think actually pretty well on these schematics here, is that this mouse brain looks fairly simple from the outside. It's kind of smooth and there aren't very many hills and valleys, whereas the human brain is very sort of lobulated and folded. We call those hills and valleys of the brain sulci and gyri. And mouse brains do not have sulci and gyri. The other thing that is different between, or another thing that's different between human brains and mouse brains is the human brain contains a much larger system of cerebrospinal fluid inside it, the ventricles or spaces which contain the spinal fluid, and mouse brains are very, very tiny. And the part of the brain that is much more complex, this folded part on the top here, is what we call the cerebral cortex. So when you yourself are thinking about your brain or the brain of your child, what we usually think about as sort of the functions of the brain is really done by the cerebral cortex. So the thinking part of your brain, the seeing part of your brain, the talking part of the brain, your hearing part of your brain, the part of your brain that allows you to move your body and feel the world and smell. All of those parts are mainly performed by the cerebral cortex. And all those folded areas that are much, much, much bigger proportionally in the human brain than the mouse brain are in that area. So that's just sort of a little bit about what the differences are, literally just anatomically, between the human brain and the mouse brain. Okay, so hopefully you're not too surprised that a mouse brain isn't the same thing as a human brain. But why then should we study human epilepsy? If you're coming to the seminar today, you're probably already interested in epilepsy, but just a few points. There have been quite a few estimates of how many people have epilepsy. Current estimates are about 50 million people in the world, including 10.5 million children. 20 to 40%, depending on the type of epilepsy and depending on the study, of people who have epilepsy will continue to have seizures despite the very best, newest, greatest medications that can be offered. And for those patients who continue to have seizures after medicines, some of those patients are eligible for surgery, but in the very best case scenarios, those surgeries will offer about 65% to 90% of chance of achieving seizure freedom. You know, what we would like to be able to tell you, especially I would like to be able to tell you as a neurosurgeon, is that we have different treatments that are all above 90% chance of seizure freedom, but we're not there yet. So there's still places for us to go. Of course, that doesn't mean that surgery is not a good option, because if you compare that to the outcomes for just adding another medication, they're much better. But we always want to improve, and this is one of the reasons. And then, as I'm sure you know, children with epilepsy who grow into adults with epilepsy have high healthcare costs. They have increased incidence of death, intellectual disability, depression, and really many other disorders. So it's a disease that deserves to be studied. And so that is why I want to study it. All right. So what are some of the difficulties that we may have when we're trying to develop new medications or other types of treatments for brain disorders? The first is one that I actually still struggle a little bit with when looking at designing therapies and you know, having potential clinical trials is the difficulty in judging the outcome. Now that may seem a little silly because you think, well, if the patients have seizures, you should see how many seizures they're having before and after the treatment. If the patient becomes seizure-free, the outcome is obvious. However, there are some children, as you all well may know, who have so many seizures per day that is almost impossible to quantify how many seizures they're having. And so if you were, say, to have a 50% reduction in seizures, it may be hard to tell whether the patient who was having 100 seizures a day is now having 50 seizures a day, or is it really 60 seizures a day? And that's obviously a big difference. Some treatments for epilepsy also can take a long time to start working really well. So that can be a difficulty. Another difficulty is the long time of preclinical testing. On average, medications that treat brain disorders have been tested in the laboratory for at least 6 to 10 years before they even go to the first stop of an FDA trial. And then the FDA trials can last up to 8 years. And then even once that's done, it can be another year for approval. So this is all, if you add that up, 10 years plus 8 years plus 1 year, that's almost 20 years from someone having like the most brilliant idea ever to cure epilepsy and that idea actually making it to a patient. The other problem is that for medication candidates, only 8% of drugs make it through that have made it to the FDA stage, compared to 15% of drugs overall, which is probably mostly due to side effects, which again, you're all familiar with side effects of epilepsy medications, because the medications are acting on the brain. They also they tend to have brain related side effects. All right. What if we did want to look at brain tissue and do epilepsy research? What could we do? What were the opportunities that we could be? So the most exciting avenue is to use the brain tissue. So here at Children's, we do epilepsy surgery. Both Dr. O'Neill and I do epilepsy surgery. And some of the surgeries that we do, we actually take out brain tissue. This is brain tissue that is leading to seizures. And so it does sound a little dramatic to be taking out brain tissue, but the outcomes in this type of surgery do tend to be pretty good. So we're actually able to look at the brain tissue itself. And, that's, I find very exciting because there are really not that many other types of surgery in which we get brain tissue. And it's not something that somebody would volunteer to donate as a sample, because that's obviously would not be a safe or ethical thing to do. So we're able to get something that is very rare, and to me, precious to study. We can also use brain tissue from brain banks. Patients do donate their brains sometimes to brain banks, but the tissue from the operating room is much more valuable because it's still alive and it's from patients whose epilepsy we know a little bit about. We can also use blood, cerebrospinal fluid, and skin. And one of the things that the blood, cerebrospinal fluid, and skin can be used for is genetic studies, and we can talk about that in a little bit as well. So challenges to use human tissue. I have to say I've been pretty lucky here at Children's Colorado and CU in that obtaining the appropriate approval has been to do this in an ethical and proper way has been fairly straightforward. I've also been lucky in that our wonderful families and patients here have been extremely generous and willing to participate in my studies, which I just appreciate wholeheartedly. The one sort of challenge that will always be a challenge is finding control tissue, that is tissue from patients who don't have epilepsy. Again, it's not something that you can ask someone to sign up for if they're not having brain surgery. We do occasionally get tissue from patients who have brain tumors that are located underneath the surface of the brain. And the surgeon will have to go sort of through what's more normal brain tissue to get to the deep tissue, and that tissue would be discarded anyway. It's taken in a safe manner and only taken if it won't cause any damage. So sometimes we're able to get that tissue, but that will always be a little bit of a struggle, and we just work around it. All right, So that's just sort of a little bit of a background on why I do human tissue-based epilepsy research and why I think it's important to do human tissue-based epilepsy research. One thing that I forget to mention a little bit is that the other reason that I think it's important to do this research, I had mentioned the difference between the human brain and the mouse brain, and the NINDS, which is the National Institute of Neurological Disease and Stroke. That's the part of the NIH which is in charge of studying epilepsy. A few years ago, they looked at how successful, basic science experiments were in sort of what we call translating to the human disease, i.e., if we find X gene that's a problem in a mouse and we're able to fix it in a mouse, how likely are we to be able to move that to the hospital or the clinic and do the same thing in a human? And the results were not that great. And, I think there's a lot of reasons for that I could probably spend 3 hours talking about, and I won't bore you with. But one of those reasons is literally just because the human brain is not the mouse brain. And so when we're able to use the human brain to actually study a human brain disorder, the value is just super high. Excuse me. All right, so that's my little sort of soapbox on why I think what we do is important and why I really value our patients and families who are willing to donate their brain tissue to us, and why I think what we do is important. So now I'd just like to tell you a little bit about what my lab has been actually looking at for the past few years. So we study a disorder called focal cortical dysplasia, or FCD, and a couple related disorders, which are very similar, which include tuberous sclerosis, hemimegalencephaly, and all of those can be grouped into a category of disorders called malformations of cortical development. Again, the cortex is that wavy surface of the brain. And what malformation of cortical development means is that when a baby is growing in his or her mom's uterus and the baby's brain is developing, as that brain is developing, some of the neurons do not go where they're supposed to go. And so the way that the brain is formed in a small area, at least a small area in the case of a focal cortical dysplasia, is incorrect. And I'll tell you a little bit more in detail right now. So what you see here, these two pictures here, hopefully you can see my mouse. If you look at these pictures really quickly, you may not see something that jumps out at you right away. This is not something that you look at this brain and say, oh, look at that big tumor. There's something really obviously there. On the left, we have an MRI, and on the right, we have a PET scan. I think that what this FCD in this patient is actually a little easier to see on the PET scan. So if you look on the left side of the tissue, which is the right side of the brain here, you can see that there's sort of a yellowish, reddish area here. But on the right side of the picture, left side of the brain, it's a little bit green. So what that means is that in this PET scan, there's less glucose, less sugar being used by the brain in this area, which can be a sign of a seizure focus. So now that we're sort of clued into this area being the potential problem area, we can look back at the MRI. And if we look at the normal side first, so if we look over here at the left side of the picture, right side of the brain, in those folds, the hills and valleys, you can sort of see there's sort of lighter gray on the outside, and then there's darker gray on the inside. But on the right side of the brain back here, you don't see that sort of folding pattern quite as much. So again, it's a little subtle, and this is actually more of an obvious one. Some of these focal cortical dysplasias are much more subtle than this on MRI. Sometimes they don't show up on PET scans. Sometimes it takes quite a quite a studious radiologist to help us find one of these, but this is what it looks like. So FCDs are, although rare in general, they're one of the more common causes of neocortical epilepsy. And what I mean by that is epilepsy that starts in the cortex outside of the temporal lobe. So not a type of epilepsy that's caused by a gene mutation in the whole body, but a type of epilepsy that starts in one area of the cortex. And in children who undergo seizure surgery, about a third of them actually have an FCD or one of the related disorders, which I just mentioned. Patients with FCDs do often tend to be drug resistant. Surgery can be fairly effective, depending on the type of focal cortical dysplasia and how big it is. Surgery can offer between 50 and 80% chance of seizure freedom over 5 years. As I mentioned, it can be difficult to diagnose with an MRI. But one of the exciting things is that many scientists are studying these disorders and identifying genes that are associated with this disorder and identifying more and more each year. So this may be a little esoteric, so I apologize if these little dots just look like little dots to you. But if you look under a microscope and look at the features of focal cortical dysplasia, what I have here on the left is normal brain tissue. So all these little gray dots here are normal neurons. And if you squint, and I have to squint too. So if you squint, you can kind of see that there's like layers going across the top, like kind of stripes going left to right. So just sort of squint like that. Now, if we look at some tissue under the microscope from a patient with FCD, you can see, first of all, that you don't really see those lines. There's a little bit of a line on the top, but otherwise it's pretty fuzzy. And the other thing that is actually fairly obvious is that some of these dots are much bigger. So these neurons are bigger than normal. So over about the last 10 years or so, a variety of super smart scientists, not including myself, have been able to identify gene mutations in FCD in something called the mTOR pathway. So what we have here, this is what we call a protein signaling cascade, where this is a variety of proteins that all affect each other. And mTOR, which is down here, mTOR is a protein that is actually found in almost all cells of the body, and it's involved in cell growth and survival. So mTOR is actually very important in cancer cell signaling as well. But mTOR has been discovered to be sort of one of the problem areas, bad apples, in focal cortical dysplasia. So if you see, we have this big signaling pathway, and you don't need to remember any of the names of these things, but out of all of these signaling pathways here, everything in blue with a little star by it is a protein in which a mutation has been found, causing either FCD or tuberous sclerosis. And that was actually the first one that was found as a gene because, and that's the TSC1, TSC2. That was found first because patients with tuberous sclerosis actually have the gene mutated in their whole bodies, not just in their brain. So again, a whole variety of genes in this all sort of related together, which can be found to cause focal cortical dysplasia. Now, the interesting thing about FCD, though, is that unlike in TS or in some of the genetic generalized epilepsies, the mutations in most patients with FCD are not found in their whole bodies. It's something called a somatic mutation. So somatic mutation just means a mutation that's found in some cells of your body, but not all cells of your body. So you weren't born with it, it happened after you were born. which is a little bit weird that the mutation would happen after you're born. So how would that happen? So if you look at this little schematic here, we start at the left. This is basically during embryogenesis. So when the fetus is 8 weeks old, you have all these little round blobs here, which are neuronal precursors or neuronal stem cells that are destined to become neurons later on in life. And something happens for some reason, and a mutation happens in the mTOR pathway. And that's this orange neuron here. Because it's a neuronal stem cell, it is going to make more cells. That's what stem cells do. They divide and they make more cells. And because that mutation has happened, that mutation will keep happening. So as that orange neuronal precursor divides, you get more orange neuronal precursors. And as the way that the brain develops, the neurons start in one place and then they migrate to where they're supposed to go. And these orange neurons, some of them become neurons like they're supposed to, but they don't migrate properly. And some of them become weird cells that they're not supposed to become, called balloon cells. So in D, this is what an FCD would look like. And in E, this is what a nice little orderly cortex should look like. So that's how a somatic mutation can occur, and how a mutation in a single cell can lead to a dysplasia in the whole cortex. And we call that clonal expansion, where a single mutation leads to problems in many more cells later on in development. Okay. So how I study FCD, after just telling you about how important it is to use human tissue and not mouse tissue, I actually do use both. So I use parallel models of dysplasia. So I take human tissue that we're already taking out during epilepsy surgery, and we also have a mouse model of FCD, which I'll tell you about in a second. And we do a few things. One is we do physiology in living cells. So we take this is a little chunk of brain tissue that was taken out of the operating room, and we put it under a 40 times magnification microscope. And you can see here that this is the cell, and we bring it down a teensy tiny little pipette, which we can attach to the cell and then record the activity of the cell. So we can keep the brain slices alive for about 12 to 16 hours after surgery, and do a variety of experiments on the cell to see how they are responding to the experiments that we do. We also do what we call an